Today, our topic, we're going to move into fatty acids. All right, so I'm a little behind, so I'm going to speed up a little bit. We're going to start talking about triacylglyceride degradation and the fatty acid beta oxidation. Okay? All right, so again, we always come back to this metabolism map. So we finished talking about photosynthesis, how you go from photosynthesis into the central metabolites. Now we're going to talk about the part in the yellow. Okay? So we're pretty much done talking about carbohydrate metabolism, so you should be familiar with how carbohydrates are metabolized into central metabolites now. So we've done this, so give it a check. Okay? Now we're going to focus on lipids, how to degrade lipids and how to make lipids. Okay? So really, the topic of this chapter is focused on how lipids are broken down to provide energy, and then how the energy, okay, that is organic carbon and NADH, or NADPH actually, is stored in cells as lipids. Okay, so we talked about structure of lipids last semester. Now we're going to talk about how you actually make it. Right? Now, triacylglycerides, this you should be familiar with. So these guys individually, that is a fatty acid, and you have 3 of those. Okay? And you have, this is a glycerol. Okay? 所以这种东西叫做三酸甘油酯嘛。Okay? 3个脂肪酸接在一个甘油身上。Okay? 这个东西叫甘油。Right? So, first thing first, triacylglycerol or glycerides, doesn't matter, TAG. Triacylglycerides are absolutely insoluble in water. Okay? Because if you look at the structure, it's pretty hydrophobic, right? You don't have charge, you don't have polarity. It's pretty hydrophobic, right? Even though you do have some oxygen atoms here, but they're carbonyls, so not much going on. And because the hydrophobic tails down here, this thing essentially is hydrophobic, okay? All right, so average mammal contains about 5 to 25% of its body weight as lipids, okay? And among that, 90% are triacylglycerides, okay? So it's a huge percentage, right? 5 to 25%. 所以你要看那个彭于晏那种有没有？那大概就是 5%, okay? 那如果你看像我这样的话，那大概是 30%, okay? 所以每个人不同啦， okay? 所以一般来说体脂肪大概是在 20% 上下。那你又有男生跟女生的不同 ，okay? So roughly speaking, 20%, right? But you know, if you like train yourself and you become very buff, okay, 很大一只有没有？那你可能就主要会是肌肉比较多，那你脂肪就比较少。那其实那个脂肪比较少的话，冬天就很容易冷，有没有？因为那个 heat transfer 比较快，然后就很容易冷。Okay, so in animals, your triacylglycerides are stored in specialized cells. These things are called the adipocytes. Okay, adipocytes are cells. If you have a bunch of adipocytes, we call them the adipose tissue. Okay, those are the white stuff that you see. Actually, there are 2 types of fat cells. There's the brown fat and the white fat. Brown fat is used to keep us warm. Okay, so it's essential. The white fats are the energy storage. Okay, so if you really enjoy烧烤啊，或者是这种火锅，有没有？你去吃那个什么雪花牛啊，那里面都是 adipose tissue， 那个白色的 part. Well, I mean, obviously it's white because it doesn't have as much of mitochondria and that kind of stuff. Okay, so that's why it's white. Okay, it's white cell. And since triacylglycerides, they're not charged, so it's almost entirely hydrophobic. So inside of these adipose tissues or adipocytes, they turn into globular structure like balls of fat. Okay, balls of like oil, okay, oil droplets, right? Make sure you understand, make a note, not all organism produces triacylglycerides. For example, E. coli doesn't do that, okay? E. coli does not produce triacylglycerides, right? So energy density of triacylglyceride is obviously very high, okay? Why? Because triacylglyceride is hydrophobic, so therefore it does not contain water in its mass, okay? Versus, on the other hand, carbohydrate storage like glycogen, okay? Glycogen is super hydrophilic, sorry. It's very hydrophilic because it's, sugars, it has a lot of OH groups, so it draws a lot of water. Therefore, in glycogen, for every gram of glycogen, you attract 2 grams of water. Okay, so glycogen is not, a suitable long-term energy storage. It's only used as a short-term energy storage. Why? Because number one, it attracts a lot of water. Okay, so it doesn't make sense for you to be super heavy and most of it is water and you can't use it. Okay, because water doesn't have energy, right? I hope you guys understand that. Water doesn't have usable energy for biology, right? So they are short-term energy, okay, because if glycogen can attract water, meaning what? Meaning enzymes are very close to glycogen, right? So, makes sense. Okay, so glycogen 因为可以吸水，所以 glycogen 可以泡在水里。那这个水里面就可以有 enzyme， 那随时可以很快速的去解决掉 glycogen， 把 glycogen 降解掉，然后变出 energy 出来。Okay, now on the other hand, lipids doesn't have to do that. Okay, lipids is hydrophobic. Okay, so they don't have water surrounding it. As a result, liberation of fats usually is slower. You need to have specialized enzymes with specialized machineries to access the fats. Okay, but because lipids do not have water surrounding it, like soaked in water, so the density is obviously higher. This is very obvious. Okay, so many animal tissues can use energy from triacylglycerides. Okay, one exception is the brain. Okay, which highly desires glucose. Okay, so last semester we talked about how the brain only eats 2 food, okay? Glucose and ketone bodies. Right? 所以我每次在说那个，像我一定要喝些那种甜的东西嘛，戒口。原因是因为就是你喝甜的东西啊，其实你会 feel happier， 因为你大脑其实 receptor 就是告诉你说它想要甜的东西，因为大脑只吃2个。glucose 跟 ketone bodies， 那其实你让它长期吃 ketone bodies 的话，你会变得很 anxious， 很暴躁，你会心情非常非常不好，然后你会很容易就是易怒 ，OK， 所以那是 ketone body。那 ketone body 的合成可以从 acetyl-CoA 去，我们到时候会讲，但是brain usually only wants glucose， right？ So triacylglycerides, pretty much every other cells can use it except your brain, right? So if we look at the tissue fuel in terms of energy density, what you can notice is that the energy content, okay, obviously your triacylglycerol have a higher amount of energy per mass compared to everything else, okay, compared to proteins, compared to glycogen, compared to glucose, okay. So unfortunately, if you want to lose weight, okay, it's actually quite difficult. You have to start from your diet. Okay, the reason for that being, if you only starve yourself, 如果你就只是不吃东西，有没有？你如果要减重，然后你不吃东西的话，其实你的身体会先开始吃糖类嘛 ，glycogen， 然后开始吃蛋白，然后最后才会吃到脂肪。所以其实不能靠那样子去减重。那如果说你是要靠重量运动 ，OK， 做那种 weight train， 那其实很多时候也不见得会达到减重啦 ，OK， 因为肌肉的 mass 的 density 会比脂肪来得高，所以很多时候你说去 work out， 然后 work out 半天，你其实没有在减重，因为肌肉会更重，只是你会看起来 nicer，OK， 就是你的 body 不会就是软的 ，OK， 所以但是你不见得一定会减重啊 ，OK， 但是that's just how it is，OK， okay? So obviously energy is stored a lot in triacylglycerides, so that's my point. Okay. All right. So first, let's talk about how we utilize it. All right. This chart is sort of a overall schematics of what we do with the fats that we eat. Okay. So if you go to have a barbecue and you eat a lot of fat, this is what happens. Okay. So the fats that's ingested in diet goes into your stomach, and your stomach will secrete sort of this thing called bile salt. Okay, 叫胆汁 okay, bile salts. And these bile salts, okay, will emulsify. Emulsify meaning they will sort of capture the fat and make it sort of like soap, right? Bile salts are essentially soap, where your triacylglycerols or glycerides are hydrophobic. Okay, so when the oil come in, they are pretty hydrophobic. And then you get these bile salts that come in, and they're going to bind to these hydrophobic droplets and turn them into like hydrophilic, and you'll see why in just a minute. Okay? And once it becomes hydrophilic, kind of, you can have enzymes doing reactions on it. Okay? So you'll break the triacylglycerides into individual fatty acids. Okay? Individual fatty acids in your small intestine, 小肠. Okay? And then your individual fatty acids can then penetrate the mucosa of your intestine, so like the 小肠 wall, 小肠的那个 membrane, and you can pass through that. Once you pass through your intestines, it will actually get reassimilated into triacylglycerides for ease of transportation, I guess. And it gets reassimilated into these oil droplets, and they form into lipoproteins. And then lipoproteins go into your capillary, and once it's in the capillary, you can transfer that into different tissues. Okay? Don't worry, I'll explain this. Okay? Now, first thing first, bile salts, or a lot of times it's called bile acids. Why it's called acids? Because this is the acid group. Okay? Why it's called salt? Because a lot of times it will represent itself in a salt form. Right? And bile salts or bile acids, they are very important because they have both hydrophobic and hydrophilic characteristic. Right? Why is that? Because the molecule is very extensively packed with carbons and hydrogens. However, it does have some hydrophilic characteristics, and so happens that when the molecule is sort of just flying around, it has a hydrophobic phase, this side, and a hydrophilic phase, this side. All right? So when you have TAG in a solution, for example, in your intestine, okay, so this part is absolutely hydrophobic. Okay? So then the hydrophobic phase will interact with it. However, it turns the other side, this side, hydrophilic. Okay? So once the other side is hydrophilic, then it can start attracting water. If it can attract water, then it can attract enzymes. Okay? So always remember, enzymes are dissolved in water. Okay? And then what you'll get, okay, is you will get this thing called pancreatic lipase, okay, or just lipase in general. Okay? Lipase is an enzyme that hydrolyzes an ester bond. Okay, so they can hydrolyze ester bond of TAG, liberating free fatty acid and glycerol. Right, so in this little picture shown. Okay? Now, the way that the lipase works is actually very interesting. So you see how this pancreatic lipase, they get associated onto the surface of the bile salt, and then you extend a little arm into the hydrophobic part, and then you touch the glycerol and the fatty acid, hydrolyze it. Okay? And if you hydrolyze it, then fatty acids can come out, and then glycerol can come out. Okay? So this is how you degrade them inside of your intestine. Okay? Bile salts plus lipase. Right? And then the product of the pancreatic lipase will reform TAG in mucosal cells. That's crazy, I know. Right? Why do you degrade it and reform it? Okay? But that's just how it is. All right? And so after it penetrates the 小肠的那个膜啊，那个膜以后, okay, you're going to reform TAG, okay, so that your cell can carry it easier and distribute it inside your cell, inside your body. Okay? Now, these TAG are going to bind to these things called apolipoprotein, whatever. They form lipoproteins. OK， 大家对 lipoproteins 这个东西有没有印象？有印象对不对 ？OK， 你去医院啊，你去检查那个验血有没有？通常会帮你如果你去做健检，当然你们可能还 too young， 所以不会去无聊没事医院做健检。医生会量什么？血糖啊，血脂肪啊，三酸甘油脂那种东西嘛。然后还有什么胆固醇嘛 ？OK， 那不是有个东西叫什么 LDL 跟 HDL 吗？好胆固醇跟坏胆固醇 ，OK？ 那其实那些东西在讲的就是这种东西，就是这个 lipoprotein，OK？ 待会给你看。Now, so your triacylglycerides binds with the apolipoprotein, and then they form functional lipoproteins. Okay, so these lipoproteins are essentially a mixture of proteins and triacylglycerides and cholesterol. Okay, so it's a mixture of stuff. Okay, they're packed with stuff inside of these oil droplet, right? And typically they first form this thing called the chylomicrons, right? And this thing will go through your capillaries, okay, 你的微血管, and then it's going to distribute your lipids to cells, okay, adipose tissues or myocytes or whatever cell that it goes, okay. So chylomicrons can move through lymphatic system and the bloodstream. Okay, so they essentially travel through blood. And once it gets to your endothelial cells or whatever cells that can sort of store this guy, this is inside the capillary. So this is the bloodstream, and then this is cell. Okay, and what happens is your chylomicron or these oil droplets, okay, this big lipoprotein will get attached to your endothelial cell, and then in which your endothelial cell have these lipase again, so the lipoprotein lipase. So these guys can then hydrolyze out triacylglycerides into free fatty acids and glycerol, and then these free fatty acids and glycerol can then enter the cell and be either utilized or converted into TAG again for storage. Okay, remember, TAG is a big, okay, each individual TAG is not necessarily very big, but because they're hydrophobic, they come and aggregate into a big oil drop, okay? And this oil drop cannot possibly penetrate membranes. 合理吧？因为你很大的一个 oil drop， 你怎么可能就是 cell membrane 然后直接吃进来？吃不进来。所以你的 cell 里面不是会储存 TAG 吗？所以它会先把你的在细胞的外面先把那个 TAG 水解掉，变成 fatty acid 进来以后，再把它储存，再重新组合。所以 TAG 它在我们的打野的过程中，它会不断的分解组合、分解组合、分解组合。OK， 那主要原因是因为它如果是在 TAG 的 state， 它一大坨，因为它是 hydrophobic， 那它这个 hydrophobic 的这一坨东西，你很难去 do anything to it。OK， 就是它不能 cross membranes， 因为它太大一颗。Okay. Alright, so this is a table of some of the common lipoproteins found in animals. This is a table from your book. And what you notice is that you have the chylomicrons, so this is the primary lipoprotein. And then you got these VLDL, IDL stands for intermediate, and you have this LDL and HDL. Okay, so this is the, good 胆固醇, you know? 你去医院的时候你会看到 that's the good 胆固醇. And then you got this LDL, the bad 胆固醇. So you can see what they're different. They are categorized into LDL, HDL based on their density. Okay? This stands for low density lipoprotein. We'll see that in the next slide as well. Okay? And what you can notice is that LDL has a lower protein content compared to HDL. Okay? So it has obviously lower density because your lipid content is higher. Right, so the way it works, it's like this. Right? So this is the overview. Your dietary fat and cholesterol goes into your intestine, okay, where they are converted into chylomicrons. Okay, so we talked about that process. Okay, so TAG, you have the lipase here, and then comes out, and then reassimilate to TAG. TAG comes out. Okay, and then these chylomicrons can then be transferred using capillaries to essentially different places. For example, to restorage in adipose tissue or be used in beta oxidation by peripheral tissues. Peripheral tissues are like your arms and stuff like that. So they can be utilized. However, it's not possible to completely consume your lipoproteins. So what you get is the leftover. Okay? But what you notice is that the triacylglyceride content decreased. Okay? So a lot of the fats are transferred to other cells. However, the leftover proteins and cholesterol are then maintained inside of this little ball. These things are then going to go to the liver, where liver is going to do a lot of stuff. 所以真的人家说那个肝要是不好，人生是黑白是有道理的。Okay? 肝真的是 do a lot of stuff for you. Okay? It converts like lactate to glucose. It converts stuff to glucose. It also has to deal with fats. Okay? It has to deal with a lot of stuff. Okay? So if your liver is not good, then we have problems. All right, so liver. And at the liver, what you can do is then you can do biosynthesis of fats and cholesterol, sort of you process them together, and then you will reassimilate these things called VLDL, similar to chylomicrons, but obviously slightly different in terms of lipid content. Okay, you see how the lipid is smaller than chylomicrons, and VLDL again can go through the capillary and then passes its triacylglycerides to your adipose tissue or to your peripheral tissues, okay, for utilization. And then you again have these leftovers, okay? And however, sometimes in your leftovers, you have these lipoprotein lipase that can convert your IDL to LDL, okay? But regardless of it's either in IDL or LDL state, doesn't matter, okay? What they're supposed to do is then again go back to the liver, okay, and then come out as VLDL. Okay, so the whole thing is a cycle, right? Now, of course, sometimes you have these LDL or these peripheral tissues that can sort of convert your LDL, okay, and then consume the cholesterol and then spit out these HDL, okay? And what you notice is that these HDL has very little fat and mostly proteins. And what they can do is they can pick up cholesterol, they can pick up a lot of other stuff. Okay, this is why they say HDL is good for you. Right? So inside your body, if you have higher content of HDL, they can, you know, clean your blood and stuff like that. But if you have a lot of LDL, you know, you got a lot of this, they are likely to get trapped on the wall of your bloodstream of your 血管，对，所以 LDL 通常会导致那个血管阻塞嘛 ，OK， 然后 HDL 比较好 ，OK， Alright, this is the overview of lipoproteins. Alright, so we'll continue our next class to talk about the mobilization of stored triacylglycerides. Okay, how to mobilize it, how to use it, and then we'll talk about how to degrade fatty acids into acetyl-CoAs, into ATP.